Yeah,
So it's with enormous pride that I introduce someone who probably doesn't require much introduction here at Rutgers, Dr. Joe Marcotrigiano, who was a member of the Department of Chemistry and Chemical Biology, had a lab next door in the Center for Advanced Biotechnology and Medicine before becoming transiently a Howard Hughes Medical Institute investigator and then going to the NIH, where he's a faculty member in the National Institutes of Allergy and Infectious Diseases. So I met Joe, I'm just trying to think when it must have been, maybe 1996, 1997?
1995.
1995. And he came to my office to interview for a PhD, as a PhD, prospective PhD student at the Rockefeller University, where I was a Hughes investigator at the time. And I think that the reason that he came to my lab was because I made the effort to try to pronounce his name correctly.
Yep.
I think that was actually that was the only qualification that differentiated me from other excellent investigators who are also Hughes investigators, both at Rockefeller and Yale and places like that. So Joe was a BSc student in chemistry here at, or bachelor student here in chemistry here at Rutgers, and was strongly advised by the late Aaron Shatkin to do PhD studies at Rockefeller. Aaron had been a PhD student in the second class of the graduate program at Rockefeller. I think another prominent member of that class was David Baltimore,
Correct.
who went on to win the Nobel Prize for retrovirus biology. So Joe came to my lab and he embarked on a project that interested him because of the connection to Aaron Shatkin to understand how the cap binding protein in the eukaryotic translation machinery recognizes the distinctive 5' end of messenger RNAs. He succeeded admirably in that work and then went on to do some additional co-crystal structures looking at how other components of the translation machinery interacted with the cap binding protein as it interacted with the mRNA cap, and did a wonderful PhD with me, papers in Cell and other high profile journals. And then he went on to work for Charlie Rice at Rockefeller, who went on to win the Nobel Prize for his work on hepatitis C virus. And Joe reinvented himself as an infectious disease structural biologist. So it's only fitting he ended up at NIAID. Now, the subject of his talk today has nothing to do with infectious disease, of course, but it is of vital importance to understanding human cardiovascular biology, and may well open up opportunities for discovery of new cholesterol lowering drugs. So, Joe, I'm very, very happy that we're able to accommodate you in this way, and look forward to welcoming you back to Rutgers at some later date when you will no doubt have gone on to yet bigger and even more impressive structures. So thank you again.
Thank you. Thank you, Stephen. Thank you for the invitation and for your patience and persistence to allow me to give this talk. I am terribly sorry I can't be there today. I was really looking forward to seeing everyone again, and I hope to do that in the not too distant future. But anyway, yes, so today I'm going to speak about apolipoprotein B and bound to the low-density lipoprotein receptor. And let's see. Oh, there we go. Okay. So as many of you know, and Stephen sort of alluded to, I've been spending many years at this point working on hepatitis C virus. And hepatitis C virus is a very interesting virus in the sense of we know very little about it. So it's a member of the flavivirus family. I'm just showing you some pictures of flavivirus. This is the cartoon on the top left. This is the cryo-EM reconstruction on the bottom left, and you're seeing some of the high resolution crystal structures that we have for this is I believe dengue. But HCV is sort of these electron micrographs across the top. It's very unusual. It doesn't really look like a normal flavivirus. And so it was shown many years ago that it does associate with apolipoproteins, and it forms what has been termed a lipoviral particle. And these were sort of two models that Charlie had put out many years ago as to how it would do that. So it would have an icosahedral virus that would be tethered to LDL or HDL, or it could be a single particle where the virus was embedded in the LDL particle, as shown here on the right. I think the field is moving towards this lower right model, but we still don't understand it. And so what is a lipoprotein, right? And so these are a very interesting class of proteins out there. They sort of have a hydrophobic core of triglycerides and cholesterol esters or cholesterol. And ApoB is a major scaffold protein for chylomicrons, low-density lipoprotein, intermediate density, and very low density, often referred to as LDL, IDL, and VLDL. And so we're going to talk about this one today. There's a whole family of these. They tend to, as Alan Ramali, our collaborator on this, he calls it the alphabet soup of proteins. There's A, there's B, C, etc. And so this was sort of my beginnings into thinking about apolipoproteins. This was a figure I made. I took this out of my last R01 grant that I wrote at Rutgers, where I had sort of schematically put this out. So somehow HCV interacts with a variety of receptors. One of them in particular is the low-density lipoprotein LDLR receptor through its interaction with ApoB. And so I'm going to speak today specifically about this particular area in terms of today's talk. And so what is VLDL and LDL and lipid transport? And so when you ingest, after eating, et cetera, so you're taking in lipids, cholesterol, et cetera, and that in the intestines, you get the expression of ApoB48. So what do these numbers mean? ApoB100 is the full-length gene. ApoB48 is the 48 percentage from the N-terminus. And it is expressed in the small intestines. It's a shorter form, and it is important for uptaking the lipids. It brings it to the liver, where the liver then will uptake it. And these chylomicrons are very large, and they only last for a short period of time. The cholesterol is made into cholesterol ester, and ApoB100, the full-length form, is expressed in the liver. And ApoB is thought to be, sort of important in cholesterol and lipid delivery system. And so ApoB, while it's being synthesized, will uptake triglycerides, cholesterol esters, phospholipids, and get secreted out as a VLDL. These are larger particles, and as the lipids are delivered to the periphery, it shrinks down, so the triglycerides are decreasing and the cholesterol esters kind of remain, and you get sort of a much smaller particle, and this can be then taken up by LDLR, the receptor, and brought back. The ApoB is degraded, the cholesterol and lipids are taken up again, and the receptor can recycle. So it's thought that the receptor can recycle up to about 150 times, and we'll talk a bit more about that. As VLDL becomes smaller, some of the smaller apolipoproteins like ApoE, ApoC, et cetera, can fall off, but ApoB remains associated. And then the small LDL particles can cause problems in arthrogenesis, which we'll talk a little bit about. Alan likes to refer to LDL and VLDL as a triglyceride delivering system. So it's an important aspect in sort of delivering lipids to the body. And so here's kind of a distribution, sort of size and density. What you notice is these chylomicron remnants are quite large, as well as VLDL. And again, the top half of this slide here, these are all ApoB particles. And so you have this enormous size as well as density distribution for the various particles. So these are VLDL, IDL sort of in the middle here, and then LDL, and then small LDLs is kind of highlighted here. There's other LDL particles, small LDL called LP little a, which I'm not going to talk about, but this is also containing ApoB. And then HDL is a completely different beast in the sense that these are different ApoA1 and 2, or sort of the major players on these particles. But the top half here, these are all ApoB. And so how ApoB accommodates these different sizes is not really understood. And so, but why is it called bad cholesterol, right? We go to the doctor, pretty much everybody, I think, has gotten, their blood screened, and they look at your levels of LDL and VLDL and HDL, and your doctor refers to them as bad cholesterol, LDL, right? So what happens is some of these ApoB containing lipoparticles tends to be the smaller ones, or that's what it's thought, can get large. They get into the artery wall and they initiate an immune response through TLR4 and macrophages. And then as that happens, a plaque begins to develop. And so this is bad because eventually it will occlude the artery. You can rupture the artery, the plaque can fall off and travel to other parts of the body, particularly the brain, where you can develop a stroke, or the plaque can get large enough that it completely occludes blood flow, which would be a heart attack, right? And so this is particularly bad, as you can imagine. This happens in all arteries or veins around the body. The ones that are most important are the ones that feed the heart, and they are the smaller ones. So your treatment options are kind of twofold. Statins are sort of a large class that a lot of people know. They prevent the natural synthesis of cholesterol by inhibiting HMG-CoA reductase, and that inhibition of natural cholesterol synthesis increases the expression of the receptor on the surface of the hepatocyte in this case. These have been around for a while. We'll talk a bit about them. And then a second class, which is a newer class, are PCSK9 inhibitors. PCSK9 is an interesting molecule. It binds to LDLR as well as it forms a complex with LDLR and potentially with LDL as well. And it causes the degradation of LDLR. So the way the inhibitor works, these are approved antibodies that block the interaction between PCSK9 and the receptor. And this causes the receptor to be recycled and not degraded, and then you get more expression of LDLR. So the treatment options work by increasing expression of LDL receptor, and that causes a decrease in your LDL levels. And so these are the two major classes that have been approved. But interestingly, there is no approved therapy against LDL itself. So why is this a problem? Cardiovascular disease is the #1 cause of human mortality in the world. In 2019, there's over 18 million people died of cardiovascular disease worldwide. That is more than cancer, respiratory, and digestive diseases, the next three combined. I mean, the numbers here are just staggering. So in the US, it's the leading cause of death for men, women, and pretty much all ethnic and racial groups. Every year, over 800,000 people have a heart attack. 600,000 of those are first time. A person dies every 33 seconds. So in this 1 hour for this seminar, that's 110 people. It's 1 out of every 5 deaths is caused by cardiovascular disease in the United States. And from 2019 to 2020, heart disease cost the U.S. economy $250 billion. The American Heart Association estimates that number to be about 1.5 trillion in the next 15 to 20 years. So this is an unbelievable problem worldwide. And so the deaths are not randomly distributed. This is just a map of the US showing where most of these cases tend to lie, but overall, you can see it's pretty much across the whole board. But particularly in the South, in the Midwest, you see a lot higher incidence of cardiovascular disease relative to other populations around the country, but still a huge problem in the United States. And so cholesterol's been around for over 100 years, and it has a very well characterized, it's been incredibly important to study, as you can tell by the number of breakthroughs over the last 100 years. And I'm just highlighting a few of those. In 1919, heart attacks were recognized in humans. 1943, for this particular crowd, Dorothy Hodgkin solved the cholesterol structure, the atomic structure. The biosynthetic pathway was identified in the 50s. LDL in 1955 was shown to be a risk factor for congenital heart disease. In 1973, seminal work by Brown and Goldstein identified the LDL receptor. Shortly thereafter, a couple of years, a decade or so later, the first statins were approved in the United States and I think worldwide. And then in 2015, the anti-PCSK9 antibodies were FDA approved. And so this work was, absolutely seminal in the field. At least four Nobel Prizes have been awarded. You could argue there's been a few other ones, but these are the major ones over the 100 years of cholesterol research. But the one thing that's actually missing for many years was a structure of LDL. So why is that? Not from lack of effort. So X-ray crystallography, this was in the mid to late 90s. These are LDL crystals. These are quite beautiful crystals, as you can see. The major problem was the resolution was lacking. So about 29 angstroms and about 15 angstroms. This is highly anisotropic. You can see one reflection at 15 angstroms, but those of us that have been doing this a long time, this is pretty promising, but again, far from atomic resolution. And then with the enhancement in cryo-electron microscopy and the new detectors and the technology that has been the great leaps that have been made in cryo-electron microscopy, there's been a number of groups for a long time that have attempted to solve the structure of ApoB by cryo-EM. So 2010, 11, there were some initial reports of low resolution and some recent stuff. This particular work in bioRxiv, it actually was published at the same time as us in Nature this year. But these were all very low resolution, like 9 angstroms or so. They had a portion of the N-terminal domain at about 5.5 angstroms, but we really are lacking here in resolution to sort of understand what really this looks like. And so that leads me to the challenges. ApoB is an incredibly large protein. It's 4,536 amino acids in its mature form. It cannot be produced recombinantly, so the sample has to be isolated from human plasma. It takes about 2 to 3 weeks. It takes about 2 to 3 week process to isolate that material. The sample can't be frozen, so we have to work quickly to utilize it. There's a lot of structural and composition heterogeneity. The amount of lipid per particle can vary a lot. There's a lack of feature, which we'll talk about in a moment. So the cryo-EM software really depends on having something to help it with the alignment. But LDL is basically a sphere, as you saw. And so you tend to just get a ball, a low resolution ball. And again, it's a large complex. So the one we are working with is about 2.7 megadaltons. ApoB is half a megadalton in and of itself. So there's more lipid than protein in this. We needed over 36,000 movies, yielding about 3.5 million particles. The data was just enormous. We're talking terabytes. In the end, for the one manuscript, we needed almost 100 terabytes of data, all said and done. And so this is computationally intensive, and we use the Amazon cloud for a lot of the processing, which we can talk about if you're interested. But I do want to note the size of this protein. So the entire HCV polyprotein from N to C, the whole genome is only 3,000 amino acids. So to put this into perspective on size. And so the LDLR expression is very challenging as well. And we used the lentivirus expression system that I developed when I was at Rutgers, and many of you have seen it and used it over the years. And so it's basically making a lentivirus and then using that lentivirus to transduce the cell line for expression. Um, Again, I'm just showing this because a lot of this was worked out during my time at Rutgers. And so, um, and then we use this adherent cell bioreactor for large scale production. Apparently, after talking to many people, LDLR is a very difficult protein to make. And so, but using this system, we were actually able to get about 20 milligrams per liter. So the expression was 10 to 20 milligrams per liter. So the expression was quite good. And I'm just showing sort of a schematic of the LDL receptor. So we have 7 of these LA modules. These are small domains that have a calcium coordination in the center. And there's a repeat of about 7 of them. Then we have 3 EGF-like domains called ABC, and then a beta propeller region. So structures of a lot of these portions have been available in the PDB for a while. We included a single amino acid mutation here at 193, which was to enhance stability, or else the protein does get degraded during expression. And so this was the entire ectodomain lacking the transmembrane region of the receptor. And so to add a feature to help us with alignment, we used a Lego body. So a Lego body was reported recently in Tom Rappaport's lab in 2021. And a Lego body is a very interesting. This is Tom Rappaport's lab at Harvard, developed this system to solve small proteins by cryo-EM. It's a very stable complex, and that's important because we were looking for it. The other nice feature of this thing is that it has an unusual sort of T shape with a sort of FAB on one side and a maltose binding protein on the other. The nanobody binds specifically to your protein of interest. The FAB recognizes the conserved region of the nanobody, and the maltose binding protein has a protein A and protein G motif that kind of sticks it all together, sort of stabilizes the whole thing, which is what we wanted, what we wanted as a feature. And so these were nanobodies made in camels. These have been reported previously. We synthesized them and tested them for binding by BLI, and you can see about 1 to 2 nanomolar. The receptor also bound the LDL in about 1 to 2 nanomolar, and we knew from biochemical work that the two could bind. This nanobody 4 and the receptor could bind to the same LDL. So that was all a good start. So how do we make the LDL? This work was done by a talented postdoc and postbac, Giorgio. They worked in Allen's lab at NHLBI. And so the clinical center at NIH has a blood donation portion of it. The interesting part of that is the transfusion biology at the clinical center is where Harvey Alter identified HCV through these blood transfusions that people were getting from the blood center at NIH. And he identified people who were getting hepatitis, even though the blood was non-A, non-B, didn't have hepatitis A or B. And so we went back to the clinical center and the blood center to get human plasma. A series of density ultracentrifugations were done to isolate out LDL, and then we further fractionated the LDL into sort of 3 types, trying to narrow in on the right form. The Lego body and the LDLR were expressed in my lab. This work was done in Allen's lab, and we came together to assemble the complex and then look at it by gel filtration. And so I'm showing that here. This is work that we did. What was really interesting and struck us immediately, so this is just SEC, and you can see this is the purified LDL we get from humans. And then what was fascinating was that when we added the receptor shown in blue here, the the LDL shifted over to what we thought, an earlier fraction, which suggested it was larger. And so you can see the excess, and B here is the LDL receptor. This is the LEGO body in C. So it seemed, I don't have the control here of just the LEGO body and LDL, but they are pretty much on top of each other. So something about the receptor, um, we noticed a shift in LDL. And so at first we thought it could be a multimer, or it could be that the LDL was somehow changing shape and allowed it to come out earlier. So in the beginning, we didn't really understand. And this is just the gel SDS page of this particular chromatogram. And it was a gradient gel so that we could see the small nanobody, which was less than 15 kD, with the large ApoB. ApoB always runs kind of as a smeary mess at the top. But this center isolated, the center of the peak here with the asterisk is what we used for cryo-EM. So you can see the ApoB, the LDLR, and the LEGO body in that fraction. And so here's just one of our TEM micrographs for the LDL, LDLR, LEGO body 4 complex. So what do you notice? These big round things in there, that's the LDL. And what you notice is that these very dark, dense, rich things that are sort of hanging off the side. And the other thing to notice too is these stripes in the middle. And so these stripes correlate with cholesterol esters sort of forming quasi-crystals in the middle of the LDL. So they're starting to crystallize maybe a little bit. We think they're more quasi-crystals than true crystals. What you notice is most of them in this particular fraction have 3 stripes. We can sometimes see 2 stripes and 4 stripes, but in this particular range, we see mostly 3 stripes. The crystals are thought to be, or these quasi-crystals are thought to be an artifact of cooling down the LDL. It's not certain whether these are present in human blood at 37 degrees, but there might be some. The other thing to notice too on the side is we're highlighting 2 here in particular. There appears to be what we think are dimers. And so we went through and started doing our processing. And so we're just looking at the single LDLs complexes. And so these are 2D classifications. And so what to notice here are a few things. We actually see these things sticking off the surface. Initially, we didn't know what they were, but this is the LEGO body here and shown here. And the other one is the N-terminal domain. I'll show you that in a bit. You can also see the presence of the stripes. The stripes are such a dominant feature that, I think what they're causing this to kind of smear out. So stripes are sort of the dominant feature, and it's kind of smearing out, and you get low resolution information here. But the LEGO body is helping in terms of these other views that we see. So when we go and we calculate some structures from this, we get about 7 angstroms here. And you can see these prominent features on the surface, but what you also see is this shadow here on the side. And when we split them, we can see that this is actually a dimer and this is a monomer of LDL. And so we did more work on this, and we have a mixed population of 1 and 2 LDL complexes. And so I'm showing you the one on the left and the dimer on the right. And what's important to notice, first of all, the size, about 22 to 24 nanometers. But what's interesting is this density hanging off the side. And so we're going to focus in on this particular region right here. And I'm showing the monomer in the dark gray, the dimer in the light gray. And what you notice is that the density, so there's this bridge of density that goes from one LDL to the other, and then vice versa. We're seeing 2 of these. I'll show you this in a bit more detail in a second. But there's a globular domain that's attached to one, and then on the other side, you see these ridges, what we called ridges initially. These are sort of blobs of density that are going along the surface of the molecule. And so since we had some idea of what LDL alone looked like, and we had a structure for the LEGO body previously from Tom Rappaport's lab, really the only thing that this could be that was left was the receptor. And so initially it didn't make much sense. And then we can look out a bit more. And what we noticed was that we think we had a ratio, a mixture. So we had the dimer was two LDLs, two receptors, and two LEGO bodies, whereas the monomer was one LDL, one LEGO body, and two receptors bound. So when we did this, we swamped it with three times more receptor. And so we thought, well, we're really going to drive it to binding. But what we ended up doing was kind of having a mixture of these monomers of monomer LDLs and dimer LDLs. And so you can really see the connection between the two particles in this view. And so here we go. We're looking at a bit more detail. We'll come back to this a bit later, but just showing you sort of the dimer here and the connection. So this is one receptor here across the top here, and the other one is on the bottom, across the bottom here. And so we'll talk about this. But for the moment, we're going to focus on the monomer because the monomer went to higher resolution. So out of our 700,000 or so particles, we were able to get 4.6, and then we started splitting. And this was work done by Altara Dearborn, a staff scientist in my lab. And really, Altara did an absolutely excellent job of kind of teasing all this apart. We have at least 6 classes, 0 through 5 here. And what you're noticing here is they're all around 7 to 8 angstroms. But what you notice is that there are certain features that some particles have, some classes have, and others don't. And they're highlighted by these squares and rectangles. So in this case, this one's lacking it, this one's lacking it, but this one has it, this one has it, this one has it, this one doesn't. This one does. And then you'll notice that the other feature is sometimes present or not present. And so we have different classes. You also see within the particle itself, sometimes we have regions that have no density, regions that have something present. And so we have sort of a mixture of classes. And what I'm kind of showing here is that some work Altara did, that local refinement using CryoSpark, and really just focusing on specific areas. So if we look right under here, this was to about 4.1 angstrom when we do a local refinement just around this particular area. But I think what Altera ended up doing was kind of figuring out, okay, we have all these different classes, but if we just focus them, so on these classes that have this sort of feature on one side, if we pool those together and look specifically at that feature, do a local refinement around those areas, you can see she's able to get much higher resolution. And so we can begin to sort of tease this apart and put it back together. This was an enormous amount of computation and work to sort of tease all this apart. But again, we could use non-uniform and local refinement of the different pools to get at the various regions that we were interested in studying. And so ultimately, for the monomer, we have 4 structures. So we have the full length to about 5.4. We have what we call binding site 1, which is the LEGO body, and this is part of the receptor, the ridge, that we called in the first few figures ago. So this is about 4.1. This is the second binding site, we call binding site 2, and this is about 4.8. And then if you focus just underneath the receptor here in the LEGO body, this you can see really the best resolution at 3.7 for ApoB. You can see these, I hope you can see these little tubes here, which represent the beta strands. So we can actually fit in the beta strands here. And we do have side chain density in certain regions. So we are approaching, close to approaching atomic resolution here. This is the highest resolution for ApoB in 30 years. And so we're very proud of that. So now how do we build this in? All right, the N-terminal domain here, which this is work from Jerry Sechrest many years ago, he kind of had an organization based on secondary structure prediction. So he had a mostly beta alpha portion in the N terminus. He called this beta alpha 1. Then it went mostly beta, mostly alpha, mostly beta, and then mostly alpha. He called this the alpha 2 and the alpha 3 region. So it was very segmented after the N terminal domain. And so AlphaFold 2 was the latest and greatest at that time. AlphaFold 2 could not do the entire protein, but we ended up doing parts and overlapping various fragments and I'm just showing some of these here. This is the N-terminal domain. We tried, we kind of had an idea of where the LDLR was binding. So we had various predictions doing a lot of AlphaFold to help us out here. But what you're noticing is that it's really kind of trying to make a hydrophobic core. And it was a lot of work to sort of uncoil this and dock this into our maps, which I won't talk too much about. The other thing that we had an initial problem with was how to determine the register of the ApoB. And what was obvious, even at low resolution, was that we could see the glycans sticking off the structure. They were very, very prominent in the density. And I'm just showing this one's on the this one's on the, I think on the N terminus, and these were on the belt themselves. So we could use this as a marker, and we knew based on some previous biochemistry that had looked at the mapping. So we could properly orient, or at least get it close into the map based on these glycan sites. So these were really critically important. And so I'm just going to show you, this is the 5.4 angstrom map with the full protein in it. I'll go through it. The map itself had to be downsampled a bit so that the movie would run. Hopefully the movie runs. Let's see. Oh, there we go. Okay. This is the N-terminal domain in blue. This is the beta propeller of the receptor here in this light pink. We'll talk more about this N-terminal domain, but what you notice is that the protein makes this belt sort of along the meridian of the particle. After the N-terminal domain, it goes starts making these beta strands here, and every once in a while, it will shoot out a loop and then come back. So you have this mostly beta sheet, one big loop, another loop, and then comes back into this beta sheet. There's 128 beta strands across the whole meridian. What to notice is that there is no tertiary structure except for right here. This is the alpha 2 region sort of jetting out, coming across. This region here, which we have density for, was not very well predicted by AlphaFold. We didn't really know how to model this in, so we took a very conservative route and sort of left it unmodeled for the moment. Resolution, what you see is this alpha 2 region comes across and contacts one of the loops on the other side of the belt. And so this has been termed a beta belt by Jerry Seacrest and others. And what to notice here is that there's this very short region in the C terminus of the alpha 2 region that actually forms a tertiary contact. Otherwise, it's basically a single secondary structure in depth across the molecule. Now we're going into, this is the maltose binding protein of the LEGO body. This is nanobody 4. The interesting thing about the nanobody is it recognizes the beta sheet, not a loop. And the receptor is shown in this pinkish mauve color here. And so the receptor actually tucks up right next to the LEGO body, which probably helped with stability of the complex. But what's interesting is these are the LA motifs on one side, and then there crosses over between 4 and 5 to the other side of the belt. Fascinating that it starts off on one side, crosses over, and goes to the other side. Why it does that, we don't know. And then this is the belt. Again, we reach another portion where it has a big loop out and back, and then finishes the belts. This is the C terminus in red underneath the N terminus. And then on top here, you'll notice this is the alpha 3 region. So the alpha 3 and alpha 2 regions are like your arms kind of grabbing a beach ball, where your body is the belt and your arms are kind of the portion that are wrapping around the ball. And again, we had decent density in alpha helices through here, but again, it really sort of falls apart this region. So again, a bit of a conservative build here, but trying to sort of really emphasize the regions that we can with decent prediction. And again, the AlphaFold prediction for alpha 3 and alpha 2 was quite weak. And so this is just showing in a bit more detail the overview here. So you start out at N, you wrap around in a counterclockwise in this direction. Here's the receptors labeled out. Here's EGFA, and then this would cross over into the dimer. And then this is the beta propeller on this particular molecule. And then we rotate 90 degrees, and you can see the belt in profile, as well as this is the alpha 3 region and the alpha 2 region here. What we've done in this particular cartoon is kind of unwound the density and sort of flattened it out. So this is the beta alpha 1 region. This is the beta 1 region. And you can see we have we call them panels. This is mostly beta. And then this unusual sort of loop out and then loop back. And then a second panel, which is also mostly beta. And you have another loop out here, a third panel, a fourth panel, a fifth panel, and then the alpha 3 region. Then we've mapped the LA motifs here, 3 through 7 on the structure here. The glycosylations kind of wrap around. This is the ApoB glycosylations. They kind of wrap around the receptor, which is interesting. We don't see, there's one other glycan here in the N-terminal domain, but most of them tend to cluster around the receptor binding site. And so very unusual structure. So the N-terminal domain is really the only part for which there was some structures within, remote structures within the PDB. And there were two samples, two available. So MTTP, which is a microsomal triglyceride transfer protein. And then there's the lamprey lipovitelin on the right here. They're very similar. All of these proteins are thought to be involved in lipid transport. So, but what to notice here is that they have the overall the same structure, but the ApoB N-terminal domain is more open and splayed out, whereas the other ones are much more compact and closed. And this kind of really does ride on the surface of the lipid on the particle, whereas these guys are much more closed, and you can tell by the RMSD they're not particularly close. But very interesting structures, and we don't fully understand that part, but how that works, how the transport works. And so I'm just comparing here the different regions. So this is the alpha 2, and again, these are our 6 major classes that Altera has identified. And I'm just showing you the alpha 2. We're looking down the alpha 2, so the LEGO bodies are up, and you notice some of them don't have a very good LEGO body densities. Some of them do. But the alpha 2 region here is there's sometimes you have good density for it, sometimes you have nothing for it. And so we think that the alpha 2 is kind of just floating on the surface, and again, kind of wiggling and shaking on sort of gliding on the surface of the particle. And so sometimes it does cross over and make contact with the other side, and then sometimes it's just very disordered. Why that, we don't know yet. And then this is the alpha 3 region, same thing. So sometimes we can see the decent alpha 3 region density here and here. A lot of times we don't, so that's here and here. Sometimes you get a bit partial here as well as here. So I think from our analysis, we would say the belt, the receptor binding, the nanobody binding tend to be the most stable portions of the protein, and everything else kind of wiggles and shakes a lot. And so it's understandable why the resolution for this has not been very good, and continues to be a problem. And so just to talk more about the receptors, so this is the N-terminal domain bound to the beta propeller of the LDLR. So this is the alpha beta region of the ApoB 100, and then this is the beta barrel of the receptor. You can see the contacts here. There's a pretty big surface. I'm not going to get too much into the contacts here because our resolution is a bit low, but you can see a pretty extensive surface as well as many histidines that are in the interface, and we'll talk about that in a moment. But again, decent size interface here between the N-terminal domain and the beta propeller. And so we can also see, again, very good interface at the nanobody to the ApoB here, shown in gray and in yellow. And in this sort of purplish color that's the various LA motifs. So 2, 3, 4. 6 is interesting in the sense that 6 does not make contact. It's sort of very far. There's maybe one or two contacts here, but for some reason 6 does not make contact with ApoB, which is unusual and I don't think was predicted previously. But you can see overall, this is the 4.1, 4.2 angstrom structure that we have for this region. And so what's interesting here was the mutations that have been identified. Familial hypercholesterolemia are homozygous mutations or heterozygous mutations in humans that cause an increase in your LDL. And previously there was some suggestion that the site B region would be close to the LDL receptor. This was a region of ApoB, and it was kind of close, so that was good. The other really interesting one was when we mapped all the known familial hypercholesterolemia sites on ApoB 100. They're shown here in blue, and they're all actually almost all of them are underneath the receptor. And I'm showing this one, highlighting this R350 3527, this arginine, which when mutated to a glutamine causes hypocholesterolemia, and this is found in 12% of the Pennsylvania Amish population. These kids that are homozygous have extremely high LDLs, and some of them have been known to have heart attacks before the age of 10. Some of them as 6 and 8-year-olds can have heart attacks at a extremely young age due to their high levels of LDL. And so what was really fascinating was to see for the first time that all of these map to the interface. And a lot of the ones that had been identified in LDLR also map to the interface. So the clinical and data that have been identified as well as the genetics fit nicely, very nicely with the structure that we've determined. So we're very happy with that. And so getting back to the plates here, I'm just showing that they're 3 different colors. So the yellow, pink, and the green, or I guess turquoise color here. And what you're noticing is that the ApoB wraps around on the diagonal relative to the plates. And so this is just showing you one direction, flipping it over, showing you the other direction, kind of like tying the package together. It's not tying it straight over. It's kind of going on an angle around it. And then the alpha 2 and alpha 3 regions kind of ride along the side of these cholesterol ester plates, which is very unusual. We were pretty interested in this particular aspect of how the ApoB accommodates these particular quasi-crystals of cholesterol esters, but very unusual. I don't think anybody had predicted that previously. And so let's go back to the dimer. And so once we had kind of characterized the monomer pretty well, we then went back and looked at the dimer. So we took these particles initially, expanded them out to a larger box, and then refined them to about 8.5 angstrom resolution. But you can really see nicely the dimer of 2 LDLs. And so if you used Altera, then just focused on this sort of N-terminal donor here, and could get like a 4.8 angstrom structure for that particular interface, or 5 angstrom, sorry. But here's the dimer. So we're showing 2 cutoffs here. This is a low cutoff in gray, and then the higher contour cutoff in sort of this purplish. And what you're seeing here is just a rotation of the different angles that we're seeing here. So again, the Lego bodies would be up and down in the first complex here on the left, and then this would be sort of with the receptors pointing down, and this is looking at the receptor. So here's the crossover events that we were talking about previously. And you can see this is quite a large complex, 40 nanometers across, 21 nanometers in the sort of vertical direction here, 24 nanometers here. So this is a very large complex at 8.5 angstrom resolution. And so once we had the monomer in, we could use that to sort of dock into the dimers. And again, what you're seeing here are the different LDLRs on the surface. There's two of them. And this diameter here from LA7 to the beta propeller is about 8 nanometers. From here to here is about 14 nanometers. So for this particular LDL, it doesn't seem likely that you could have a 1-to-1 ratio where both binding sites are occupied. This would have to be a lot smaller particle. So there's no way for this particle, for one LDL receptor to bind to one particle and occupy both sites. I hope that makes sense. And then if we rotate it, so we're looking on the side, the membrane helices, membrane anchoring helices would be here. So we think it sits this way on the membrane with the two LDLs sticking out. And these are C2 symmetry, and here's the axis of rotation, like right here straight off the membrane. And so Altara noticed there was obviously, we always have to deal with heterogeneity when we talk about LDL. And so we were able to see at least three different classes here. The top class, the arrows represent the direction of the cholesterol plates, and they form an acute angle. In this particular one, they form more of a 90-degree angle. And in this one, you have one with cholesterol ester and one without. When we superimpose these are all pretty low resolution, but when you superimpose these, what you notice is that the one does superimpose fairly well. The other one, ApoB, seems to be shifting over depending on how the plates are arranged. But what's interesting to note here is that the receptor doesn't really seem to mind. We think it binds all of these different factors. This particle doesn't seem to mind. It probably can deal with the heterogeneity. It must be able to deal with the heterogeneity that is present in LDL, and that allows for uptake. I mean, we need more data to get this to higher resolution, and so plus the size there's a lot of technical issues with getting this to higher resolution, but we'd like to do that. And so I just wanted to show the two together in a movie rotation. And what is fascinating here is just the juxtaposition of the two particles, right? So these are phospholipid containing particles, right? And so if they were to get this close without the protein, you would expect these to fuse. But what ends up happening is that this is the alpha-3 region that is at the interface. So the same residues come together in the two particles in the alpha-3 regions, come together and prevent the fusion event, at least in this conformation, right? So you can really see how I was struck by how close they get to one another, and they don't really, they aren't fusing together. So, but maybe that is possible if if these are getting large enough or they're high enough concentration where things begin to, you could have some odd fusion events that occur. And so here we're looking down again from the membrane up at the various particles. So this is sort of bringing it all together. Again, struck by how close these can actually get in space without fusing. So very interesting work by the ApoB 100. And so we had looked it up, and so endosomal particles tend to be about 55 nanometers. We kind of made a cartoon for how we think uptake happens here. And so there is space within a 55 nanometer circle sphere to accommodate these two. The smallest VLDL probably wouldn't fit as a dimer, possibly, but we don't fully understand that, to be honest. But you could maybe fit two of these or so in a sphere of 55 nanometers to accommodate two dimers, maybe three in this sphere. So to explain some of the uptake that has to happen in order to clear the LDL. And so during endosomal entry, there's an acidification event, and that pH is sensed by the LDLR. And so Deisenhofer, working with Brown and Goldstein, determined the structure of the low pH form of the LDL receptor. And I'm just showing here a movie, but what has to happen is that the portion that is bound to the N terminus and the LA motifs have to come together. So those surfaces that are binding the LDL surface need to, the L portions of the LDL receptor that are binding LDL need to come together. And so there's a conformational event that occurs. Here's my movie showing that movement that has to happen. So this is an enormous movement to bring the receptor together. So there would be a turning event as sort of movement, large movement event. And we believe it pivots around this EGFA region, LA7. So this part seems to be consistent between the two structures, but everything else kind of has to move to come together. And again, there's many histidine residues that are, we think, sensing that pH change or lowering. And so what about AlphaFold? I get asked this all the time. What do you get with the latest version of AlphaFold? This is I haven't checked recently, but this was when we were still in the review process. So we ran full-length ApoB, could be done by AlphaFold 3, and I'm showing the 5 predictions here, as well as the plot for confidence. What you're seeing is there is more confidence in the beta region, less confidence in the alpha 2 and alpha 3 region. But what it's trying to do is to build a proteinaceous hydrophobic core. ApoB doesn't have that outside of the N-terminal domain. Everything else kind of is a single secondary structure in thickness as it winds around. And this isn't to say AlphaFold, people look at this and they say, oh, AlphaFold is a terrible program. It isn't. It's a very good program. It's probably the best we have at modeling structures, protein structures. I think what this speaks to isn't the problem with AlphaFold, it's that it hasn't learned, and there's nothing like this before this in the PDB. And so the secondary structures, I think, were mostly okay. But again, the tertiary structures is where you really lose it. There was nothing like ApoB in the PDB. And so you get this sort of attempt to make a proteinaceous hydrophobic core, which really isn't available. There is a hydrophobic core, but in this case, it's lipid, not protein. And so the last question I always seem to get asked is how big is this thing? I mean, we're used to proteins alone, not proteins with lipids. These are sort of unusual family, a small family of proteins out there. And so what I'm showing here is just the HCV-E2 with the FAB structure that we solved when I was at Rutgers. This is the dimeric form. Again, it's 2.7 megadaltons each LDL, so it's over 6 megadalton complex here. And the closest thing in molecular weight to the LDL was the 70S ribosome, prokaryotic ribosome. And this is from Venki Ramakrishna's lab. This was bound with the 3 tRNAs, beautiful work that they did. But you can really see that basically one of these LDLs is about the same size as the prokaryotic 70S ribosome. So that kind of puts it in perspective. And again, this is protein in a core of RNA. This is protein with a core of lipid. So similar in certain, at least on a very 20,000 foot view. But with that, I kind of want to conclude. ApoB is a unique fold. Very unusual in this day and age to say that. The structure of ApoB and explains how it encircles the cholesterol ester plates. You have 2 binding sites for the LDL receptor on ApoB, leading to the formation of a dimer. And that's particularly interesting because a lot of these receptors tend to form dimers when bound to their ligands. Think like, insulin or various other hormones, things of that nature. Most of the familial hypocholesterolemia variants in ApoB coincide with the binding interface with LDLR. Stephen mentioned the ApoB LDLR complex structure may open up new avenues for intervention. These are desperately needed as people continue to get sick from elevated LDL. And then lastly, I just want to talk about, we took a patient sample and brought it to the EM for structure determination. There aren't too many samples that go from the clinic to the EM. And I've been talking to many of my colleagues here in NIH about there maybe there are other samples that we should consider taking along that pathway. And so with that, I just want to end and thank my excellent this is the group that did it. Outstanding bunch of people, excellent scientists. Mart and Giorgio made the sample, worked very hard, worked on the recombinant expression. Haotian helped prep all the grids and help with the data collection and some of the processing. Altara did all the classification. Um, just an absolutely heroic effort by all of these people to do this structure. It was nearly impossible. And I definitely want to thank Alan Raleigh, my collaborator, my friend, my neighbor. He lives around the block from me. Um, I do want to thank the Leduc Foundation for supporting us. The National Cancer Institute did a lot of the cross-linking data that's in the paper that I didn't talk about for sake of time. Our computing was all done on Amazon Web Services through the Strides initiative, which if you need computing, this also works on the extramural side. My thanks to Stephen and Kathy Lawson. When I was neck deep in building this, I kind of reached out for some support and advice. Thank you, Kathy. Thank you, Steve. And the data was collected at the NHLBI CRIOS here on the Bethesda campus. And so with that, I want to say thank you, and I'll stop sharing or take questions.
Thank you, Joe. As Eddie commented in his quick message, amazing. So are there questions in the room for Joe?
Hi, Eddie.
Jason.
I missed.
Thank you, Stephen. Thank you, Joe. What have you tried and what has worked or not worked so far in terms of new software and algorithms for analyzing the heterogeneity and continuous flexibility in the LDL particles?
Yeah, so we've been doing a lot through CryoSpark's got this new 3D variability. We've been working on those aspects of it. Just the size of the particle has been a challenge. Computationally has been a definite problem. I've tried CryoDragon. I'm getting that up, and I had a visit recently to Princeton and spoke with them, actually about a year ago, and spoke with them. I haven't tried that yet, just because, trying to get all the computation in place. This is not trivial. I think that's probably at the moment the bigger issue than getting the software running. It's getting the software running with particles of this size. I don't know if you've done anything similar, Jason, but I would appreciate any feedback on how to get that to work. But we tend to have memory issues, and yeah, it's been a challenge. So we're working on that aspect. Yes. I think Kathy has her hand up. Oh, sorry, go ahead.
So there's a question from Luigi Di Costanzo, a former RCSP PDB staff member. Great talk and effort. For curiosity, did you try to fit the cholesterol crystal structure inside the particle receptor that you're resolving?
Yes, I did, Luigi. I tried. There really isn't very good density for it is a problem. The cholesterol is we're struggling with that aspect of it. I've tried because the cholesterol, there's a couple of different crystals in the Cambridge database that we've tried fitting in there. The resolution for that is poor, and I mean, it's something I'm definitely interested in and wanting to do more like electron diffraction on the crystals to see if that might be a way to get a handle on it. But I'm wondering if the orientation of the lattice is either different relative to the protein. So if you completely sort of negate or or focus on the protein aspects and remove the cholesterol, it tends to give you better resolution. We focused on trying to get better resolution for just like masking out just the cholesterol plates. That didn't work. Yeah, I know that's a difficult part that I'm very interested in understanding more of. But yeah, so far I've come up on the short end, Luigi.
So
Good
powder
question.
diffraction might work there, Joe. I mean, you
Yeah,
can predict the powder pattern from the crystal structures that are in the Cambridge Structural Database, and then you might be able to record a powder pattern from the sample
right.
and make a correlation there. There's also a question from Hyun. Great talk. I'm interested in the maturation of apolipoprotein. I wonder if you find any difference of the ApoB structure on chylomicron ApoB48 or VLDL ApoB100 versus LDL ApoB100.
Yeah, great question. Something that we're looking into. Again, how does this ApoB accommodate, something that's 4 to 5 times its diameter, right? I mean, yeah, that's something we're definitely interested in. But we have some ideas, but at the moment they're only just ideas. We need to get some more data, and then the VLDL is very challenging because of the heterogeneity that just size-wise is. We looked at some VLDL, and it was very heterogeneous in terms of just size of the particle. But yeah, that's something we're definitely interested in.
So are there any more questions online or
I
in
see
the room?
Kathy has one. Kathy has her hand up.
Okay, Kathy, are you going to speak? Kathy Lawson, you are now good to go. We cannot hear you online. You have to unmute yourself, Kathy. Can you unmute her yourself? No, okay, all right. Okay. Can you type your question into the chat, Kathy?
I don't see the chat.
Can someone unmute your microphone? Only you can unmute your microphone, Kathy. Okay, Kathy.
Oh, interested in the residue profile inside and out. Yes, good point. I did look at it. It is more hydrophobic on the inside than on the outside, as you would predict, but it is not. There are some charged residues on the. It is definitely more hydrophobic inside than outside, which I think is what you're relating to. It is not completely hydrophobic though, um, because you probably need to neutralize the triglycerides that interact with the triglycerides and the the phosphate backbones that are there in the phospholipids, et cetera. So it is definitely more hydrophobic, but I wouldn't call it. It's not as stark as you would think, Kathy. I checked that too. I should have a picture of that.
Cool. Well, Joe, this was an amazing piece of work. Thank you very much for presenting today. We will have you back to Rutgers once the NIH lifts your travel ban, and look forward to hosting you here. You had about 30 people
would love to
online
come
and
back.
about 20 people in the room, so you had a great crowd. Thanks a lot. Good luck.
Thank you very much.
Be safe.
Thanks. Talk to you soon. Bye, everyone.
Bye.